Hello, this is your tutorial on how to use the human karyotyping gizmo, which you're going to be doing as an assignment on November 2nd in class. So you're going to be heading up to the computer lab, go ahead and open up the internet, and then go to your Google Classroom page. Yours is going to look slightly different, of course, because you're going to be in your student view, but you're going to open up the Karyotyping Partner Lab. When you do so, there will be an attachment. It's view only. It'll look like this. So what you're going to want to do is you're going to want to go to File, make a copy, and then you're going to want to put your name in it. So this would be Karyotyping Lab Report. Save it into your drive, and then you're going to want to share it with your assigned partner. So make sure you're typing their email address in and then giving them privileges to also edit the document, because you two are going to be working on it together. So this is the document that you're going to use to guide you through, because I'm not there. The very first thing it says is your username and your password. You're going to go to explorelearning.com and click login using this username and this password. It's the same for everybody. It was just way easier that way. So I'll go to explorelearning.com. I'm going to click up here to log in. My username was demo719. My password is lowercase b, blue, 719. Go ahead and hit login. It'll automatically tell you that your teacher is me, and you're in the section for fall 2016, and you're going to launch this gizmo. So what you're seeing here is a whole bunch of different chromosomes. This is actually what our chromosomes look like inside of our cells when they've been dyed and stained with different chemicals. And as you're going to do part A, the lab asks you to do yellow parts by both students looking on the same computer. Partner 1 is going to do anything in green. Partner 2 is going to do anything in blue. You guys determine who's partner 1 and partner 2. So before you begin, tell me what you know about chromosomes. Answer these two questions. Then go back to the gizmo, and it says, how does the appearance of chromosomes change as you move from chromosome 1 to 10? So back in my gizmo, I'm just going to click here, and I'm going to describe the change that I see as I continue clicking through them. I'm going to type my answer in right here. And then it says, examine the chromosomes labeled X and Y, and how do those compare? So you got to keep clicking through until you get to the chromosomes that are called X and Y. You're going to describe those and compare them as well. Type your answer in. The next part is where you're going to split up. So partner 1 is going to reset the gizmo on their computer. There's a reset button right here. There's really no need to do that, but it does take you back to 1, which is nice. And then it says, make sure subject A is selected. Do that right up here. There's A, B, C, D, and E. Make sure you're on the correct subject. And then you're going to match up the chromosomes and try and find them in the simulation. So what I'm going to do is I'm going to grab any chromosome out of my pile of DNA. I'm going to find this one. So I'm just going to click through. Actually, I'm going to send that one back. I don't like that one. And I'm going to look and see if these look like a match. If I think so, this is chromosome number 2. I'm going to click and drag this over here and see. If it stays there, that means it was a correct match. If I try this one and I try to put it in chromosome 2, well, first of all, it doesn't look anything like it. But it'll zoom back and it'll tell me I'm wrong. I got to keep looking for that one. That one's chromosome 5. So I drag it over and do the same thing there. You're going to be sorting out all of these chromosomes one at a time, putting them in the right place, making sure that they match the ones that are in here. Eventually, though, if you get enough of them filled in, so if I've got one copy of a whole bunch of them, this batch button will allow you to click on it, and it'll fill in the matching partnership when you get close enough. So don't forget to do that. That's important. When you're finished and all of the chromosomes are put together, you're going to use this camera button to take a snapshot of the karyotype. Now that I'm done, I'm going to take a screenshot of it. So I'm going to click this little camera icon. And then you're going to right click on it to hit copy. Go back to your lab report and it says paste the snapshot below, shrink it to fit this page. So you're going to use Control-V, paste on your keyboard. That's ginormous and we don't need it that big, so just shrink it down so that it still fits on the page and doesn't mess things up down here. Make sure you've got enough room that you can actually fit all of the answers to your questions. Okay? That's just to make sure that you did it right and that you're seeing the right things there. So the next thing, you count them up, answer the questions, so on and so forth. Partner B, you're going to do the exact same thing. Paste in your answer. And then you're going to go to the next part where you're both going to work together. You're going to reset and go to subject C. Match them all up. And this time you're going to find something different. So you're going to find what is that difference? Why doesn't it look like A and B? What's different about it? Once again, you take a snapshot, paste it below. And then you're going to have to diagnose them. Do they have something like this? And if so, record the stuff that's going on. Click the Diagnosis tab to find your answer. This is the Diagnosis tab right here. That's the one you're going to want to click to get the answers. Describe the symptoms of having that anomaly in your chromosomes. Then you're going to split up again. Subject D and subject E are each going to be done by a different one of you. So you go back to your lab, choose subject D if you're the first partner, subject E if you're the second partner. Sort them all out, find out what's wrong, click Diagnosis, fill in the table. Make sure that you put both of their karyotypes, their maps of their chromosomes down here, and make sure they shrink so that they fit on the page. These are just some conclusion questions here. What type of chromosomes have the greatest effect and why? How do you think sex is determined? What do you do if you have a strange scenario like 3 X chromosomes? And then the genome. If you're confused about genome, ask Mr. Stace. He's kind of an expert on this kind of material and he can talk you through genome. But what do you think you can find out by looking at one of these? And what can't you find out by looking at one of these? That's what the question is after. Once you are completely done, I've put a reminder at the bottom of every single page of the document, but you need to print the document and turn it into Mr. Stace. He'll have a stapler to put it all together. Make sure as well that your names are printed up here at the top. And that would be it. Good luck to everybody. If you have questions, go ahead and send me an email.